Welcome back, everyone. We're diving into a really cool topic today, how our DNA, the instruction manual for life, actually gets turned into the proteins that, well, build and run our bodies.
And the answer, it's all about RNA.
RNA.
Yeah. For a long time, people thought it was just kind of hanging out next to DNA.
Like the sidekick.
Yeah, exactly. But it turns out RNA is the key to the whole process. Our guide for this journey is Life's Greatest Secret: The Race to Crack the Genetic Code by Matthew Cobb.
Awesome. So where do we start with this story?
Well, the cool thing is this story brings together all these different scientists,
Like who?
physicists, chemists, even code breakers, all trying to understand this, language of life.
It's kind of like a detective story.
Oh, it totally is.
Very
Full of twists and turns, brilliant minds. And it all starts with this physicist, George Gamow.
Wasn't he the Big Bang Theory guy?
Yep, that's him.
So how do you end up in the world of DNA?
Well, after Watson and Crick figured out the whole double helix structure of DNA, Gamow saw the genetic code as, the next big scientific challenge.
So from the universe to our cells, that's a pretty big leap.
Right. But Gamow was all about tackling these huge questions, and he had a great sense of humor, too.
Oh, yeah.
The book mentions how he once added a colleague's name to a paper just so the author list would match the Greek alphabet.
Seriously.
Yeah, he was character, that's for sure. But he brought that same playful energy to his science. He was convinced math and physics could be used to solve this biological puzzle.
I like his style.
Yeah.
So what did he do?
He started this club called the RNA Tie Club, all about deciphering the genetic code. They even wore ties with RNA embroidered on them.
No way. A secret society of scientists.
Pretty much. But hold on, you might be thinking, why RNA? I thought DNA was the big deal in heredity.
Yeah, that's what I learned in school.
Okay, so DNA is definitely the blueprint, the master copy of our genes. But scientists realized there had to be something carrying those instructions from the DNA to the part of the cell where proteins are actually made.
Like a messenger.
Exactly. And RNA is found in both places, in the nucleus with DNA and out in the cytoplasm where proteins are made. So it fit the bill perfectly.
Okay, makes sense. So the RNA Tie Club figured that part out.
Yeah.
But how do they know how to read the code? I mean, DNA's got these 4 letters, A, G, C, and T, but proteins are made of 20 different amino acids.
Right. It's like trying to write a whole book with only 4 emojis. Not very expressive.
Right at all. How'd they even begin to figure that out?
Well, Gamow, being the math whiz he was, proposed that the code was based on triplets of letters.
Triplets.
Yeah, what we now call codons. Each 3-letter combo would specify one of the 20 amino acids.
So, 3 letters for 1 amino acid.
Exactly. But they still had to prove it. And that meant finding the actual messenger molecule, the RNA that carried the instructions from DNA.
Ah, so that's where messenger RNA or mRNA comes in.
Bingo. But finding mRNA wasn't easy. It's super rare in the cell, and it breaks down really quickly once it's done its job.
So how did they find it?
Well, it took a bunch of brilliant scientists, folks like Francois Jacob and Jacques Minot. They kept hitting dead ends but refused to give up.
Persistence. I like it.
Right. Eventually, Sidney Brenner, Francois Jacob, and Matt Meselson figured out a way to use radioactive labeling and this technique called ultracentrifugation.
Sounds complicated.
It was, but
Right,
they
though.
managed to isolate and identify mRNA.
Wow, that's huge. So they finally had their messenger.
They did, but the question remained, how did it actually work? Think of it like a record player.
Okay, I'm listening.
The ribosome, that's the protein-making machinery in the cell, is like the turntable. The mRNA is the record, and the protein, that's the music.
Okay, that's a cool analogy.
Right. So the mRNA carries those instructions from DNA, telling the ribosome which amino acids to link together.
Just like the grooves on a record.
Exactly. So we've got DNA, the blueprint, mRNA, the courier, and the ribosome, the music player.
Awesome. But how do we actually get from those 3-letter codons to the right amino acid sequence?
Ah, well, that's where things get even more interesting. Because there's another type of RNA involved.
Another one. How many are there?
Don't worry, we're almost done. This one is called transfer RNA, or tRNA.
Okay, key RNA. What's its role?
Well, tRNA is the superstar that links those mRNA codons to their matching amino acids.
Like a translator.
You got it! It was actually Francis Crick, the DNA double helix guy, who first predicted these little guys.
He was on a roll.
He was. He figured there had to be something connecting the codons to the amino acids.
And he was right.
Of course. But it took another scientist, Paul Zamechnik, to actually prove that these tRNA molecules existed.
So many players in this story.
I know, right? It really shows how all these brilliant minds, each with their own expertise, came together to solve this puzzle.
That's pretty mind-blowing.
Totally. But even with mRNA and tRNA in the picture, they still didn't know which codon went with which amino acid.
It's like having a map but no legend.
Exactly. And that's where our story continues. Stay tuned to find out how they finally cracked the code. So last time, we left off with this big question.
How to actually read the code.
Exactly. We knew messenger RNA carried the instructions, and transfer RNA helped link the codons to the amino acids.
But we still didn't know which codon meant which amino acid.
Right. It was like having this, secret code book, but no key to decipher it.
So how did they figure it out?
Well, that's where our next scientific hero comes in, Marshall Nierenberg.
Okay, tell me about him.
So Nierenberg, he was a biochemist and kind of an outsider in the scientific world.
The book made it sound like he wasn't afraid to take risks.
Totally. And that's what led him to make this, huge breakthrough. He figured out how to make proteins in a test tube.
Wait, what? Outside of a cell?
Yeah, he basically recreated the whole process of protein synthesis.
Wow. That's amazing. But why was that so important?
Think about it. Instead of studying this super complicated process inside messy cell, he could control everything.
Like you could just add different ingredients and see what happened.
Exactly. So he started testing different RNA sequences to see what kind of proteins they would make.
That sounds like a lot of trial and error.
It was, but eventually he hit the jackpot.
Oh, tell me. What did he find?
Okay, so he started with this really simple RNA molecule, all made of uracil. We call it poly-U.
Poly-U, got it.
And guess what? It made a protein that was all one amino acid. Phenylalanine.
Wait, so that means the code in UU.
Codes for phenylalanine. Boom!
They cracked the first part of the code.
Exactly. It was like finding the first word in a whole new language.
Wow, I bet they were thrilled.
Oh, yeah. It was a huge moment. And from there, they just kept going, testing more and more RNA sequences.
Like a giant puzzle.
Totally. But of course, it wasn't always that easy. They needed a way to create very specific RNA sequences.
And that's where Gobin Khorana comes in, right?
You got it. He was this brilliant chemist who specialized in synthesizing nucleic acids.
So instead of just finding random RNA molecules, they could actually design them.
Exactly. And that made a huge difference. They could systematically test all the different codons.
So biochemistry, chemistry, it really took teamwork.
Absolutely. And their work paid off. By the mid-1960s, they had cracked the entire genetic code.
The entire code.
Yeah.
That's incredible. They basically figured out the language of life.
They did. It was a major scientific achievement. Both Nierenberg and Khorana ended up winning the Nobel Prize for their work.
Wow, well-deserved. But even with the code deciphered, there was still this question of how it all worked. Like, how do those mRNA codons actually get linked up with the right amino acids?
Right. We had the code, but we needed to understand the mechanism.
Like the nuts and bolts of how it all fits together.
Exactly. And that's where our friend, transfer RNA, comes back in.
tRNA. Right, that was Crick's big prediction, the adapter molecule.
Exactly.
Yeah.
But how does it actually work? It's like each tRNA molecule, it has this special 3-letter sequence.
A sequence.
Yeah, it's called an anticodon. And it's like the perfect match for a particular mRNA codon.
So if the mRNA has the codon, say, EUG,
the tRNA with the anticodon UAC will bind to it.
that's like a lock and key.
You got it. And on the other end of the tRNA molecule, there's this little spot where the matching amino acid attaches.
So the tRNA brings the codon and the amino acid together.
Right in the ribosome.
Okay, I'm starting to see it now. So the ribosome is reading the mRNA codon by codon.
And the tRNA is bringing in the matching amino acids one after another.
Like a perfectly choreographed dance.
Exactly. And as those amino acids are delivered, they get linked together, forming a chain.
And that chain becomes...
The protein.
Yeah.
And the sequence of those amino acids, that's determined by the original mRNA instructions.
Wow, it's just incredible how all these tiny molecules work together.
It really is amazing. And to think, it all started with this simple question. How does DNA's code get translated into proteins?
And that simple question led to such huge discoveries.
It totally did. And the story doesn't end there. This knowledge about the genetic code, it's opened up so many new possibilities.
Like what?
Well, things like genetic engineering and personalized medicine. But we'll have to save that for next time.
So we've been on this incredible journey, right? We've seen how scientists crack the code of life, this amazing dance of DNA, RNA, and proteins. But it's not just about understanding the past.
Right. It's about the future.
Exactly. This knowledge, it's like opening up all these possibilities, like genetic engineering. We can actually manipulate genes now, right?
We can.
Yeah.
Because we understand how DNA's code gets turned into proteins, we can actually insert, delete, or even modify genes.
So we can actually, like, rewrite the book of life, fix typos, add new chapters.
We're not quite writing novels yet, but we're getting there. Scientists are already using genetic engineering to make crops resistant to pests and diseases.
That's amazing.
And even creating bacteria that produce insulin for people with diabetes.
Wow. So we're already seeing the benefits.
We are, but there's a whole other side to this too.
What do you mean?
Well, there are a lot of ethical considerations with genetic engineering. We have to think about how we use this knowledge responsibly.
Right. It's a powerful tool. We need to be careful.
Exactly. But let's switch gears a bit. You mentioned personalized medicine earlier.
Oh, yeah, that's another huge area where this knowledge is making a difference.
Right. Personalized medicine is all about using a person's own DNA to help make healthcare decisions.
So, like, tailored treatments.
Exactly. By analyzing someone's DNA, we can see if they have any genetic variations that might make them more likely to get certain diseases.
Or if they might respond better to certain medications.
Exactly. It's like having a personalized health roadmap based on your own unique genetic code.
That's so cool. But is this really happening now, or is this still, like, sci-fi stuff?
Oh, it's definitely happening now. Personalized medicine is already being used to guide cancer treatment, predict bad reactions to drugs.
That's incredible.
And it's just the beginning. As we learn more about the genome, there will be even more ways to use personalized medicine.
It's amazing to think about all the possibilities, how this knowledge about DNA and RNA can actually help people live healthier lives.
It really is. It shows how important basic science is. Understanding the fundamentals can lead to these huge advances.
Totally agree. Well, as we wrap up our deep dive into RNA and the cracking of the code of life, I'm just in awe. It's incredible to think about this journey, how it all started with a simple question.
How does DNA get translated into proteins?
Exactly. And that question led to all these incredible discoveries. It really highlights the power of human curiosity.
It does, and the ingenuity of these scientists.
And it all started with this little molecule, RNA.
Which turned out to be so much more important than anyone imagined.
From carrying genetic information to regulating genes.
Who knows what else we'll learn about RNA as scientists keep exploring?
Maybe we'll even figure out how life itself began.
Yeah, maybe. It's a question that's fascinated scientists for centuries.
And it's definitely something to think about. Well, thanks for joining us on this deep dive into the amazing world of RNA and the cracking of the code of life.
It's been a pleasure.
Until next time, stay curious,